Cloud. Hi, this is the Cancer Patient Lab, and we're excited to have Michael Castro with us again today. He's spoken to our group of patients before. He's a pioneer in looking at pathways and figuring out how to block those. So he's an unusual researcher combination with an MD because he's treating patients in the Southern California, and he's based in the Southern California area. Our standard disclaimers are two. One is that this conversation will be public. So if you're concerned about what might be said here, disguise yourself, hide your image, or change your name, or don't say anything. And then the second is that this is not medical advice. This is information for you to take to your medical team. Okay, so with those two disclaimers, Michael, you were starting to say something really interesting about what brought you here today.
Yes. In the course of my ordinary patient care, I came up with a rather spectacular realization, and I wanted to share it with you guys. And so I re-invited myself. And if I'm allowed to share my screen, I have put together a PowerPoint, which I can bring up right now.
It's working.
Terrific. So I work for this company, CellWorks, as many of you know. And CellWorks is a computational biosimulation company. It's working, except I cannot make it go forward. Let's see here. Oh, here we go. CellWorks is a computational biosimulation company that takes complex genomic information and then does signaling pathway analysis. And essentially, I want to go all the way back to 1996, where the medical oncologist's role in managing prostate cancer really began. And at that time, we were giving prednisone as a palliation. And there was, interestingly, some improvement in PSA that could be achieved from that, probably because prednisone inhibits NF-kappa B. In any case, mitoxantrone was really the first drug to come along, and it shows an improvement in PSA response, as you can see here. And the PSA response curves look like that in favor of chemotherapy. And so thus began the era of giving chemotherapy to metastatic prostate cancer patients. And in this trial, they did a very admirable job of looking at quality of life. And in spite of giving a cytotoxic drug, the argument was that patients actually felt better, and therefore it was a reasonable thing to do. There was an absence of people feeling worse, for example. On the other hand, the survival, there was no improvement in survival by adding mitoxantrone. And nevertheless, this was the standard of care for nearly a decade until this famous landmark study of mitoxantrone versus I mislabeled the slide. It's really docetaxel versus mitoxantrone prednisone. And they had about 1,000 patients who were getting docetaxel either in a weekly or every 3-weekly schedule. You can see the every 3-weekly schedule surpasses this. Of course, you had to live about 8 months in order for there to be any difference at all. But after that, there was a clear benefit in favor of docetaxel. And as we do in evidence-based medicine, we declared that docetaxel is the new standard of care. And mitoxantrone has pretty much been in the waste bin of medical oncology ever since. The 10-year follow-up supported this conclusion and enforced this as the standard of care. And today, for all patients who show up with hormone refractory disease, docetaxel is the first line of treatment. In fact, now it's given in the upfront setting with hormone deprivation simultaneously, because you get an even bigger increment Now, at CellWorks, what we do is to take the results of NGS and we input it into a computational model of signaling pathways, which can give us a picture of how the disease creates the malignant phenotype, by which I'm talking about the hallmarks of cancer. And probably this is a sophisticated group of people I'm talking to. You know what the hallmarks are. It's uncontrolled proliferation, blockade of apoptosis, angiogenesis, replicative immortality, angiogenesis, immune evasion, and so on and so forth. There are 4 new ones, by the way, as of last year. And once we create this network of disease biology, then we can simulate how drugs and drug combinations interact with the network. And because the whole thing is based on arithmetic, or actually differential equations, we get a quantitative output about how we think one treatment will be compared to another treatment. And then we get the signaling pathways that are going to show us why. And so this is what I call explainable artificial intelligence, in contrast to the normal concept of black box AI, where you give something, 100,000 data points, and then you let the computer find the associations. And in the end, you get a model and you're not quite sure why it says what it says. This, on the other hand, is much more satisfying, particularly if you're giving patients drugs based on it, because you can see what you're doing and why you're doing it. And essentially, it's, as I said, a signaling pathway impact analysis tool. And it really gets to show you the network, the complex proteogenomic network that cancer is. And this network is characterized by specific nodes, which are oncogenes, master regulators. Master regulators, of course, are involved in determining cell fate. All the cells in the body have the same DNA, but they have different specializations based on their transcriptional organization. Then we have synthetic lethal nodes, which are extremely important because normal tissue doesn't have those. And so you can kill a cancer by attacking synthetic lethal nodes without harming normal tissue, which is the opposite of how most of us were taught to think about cancer therapy, where the concept is, because of the overlap between normal human biology and cancer, there was no alternative except to cause normal tissue toxicity and go to maximum tolerated dose in order to get benefit. Well, if you think about synthetic lethality targeting, it's the opposite. You can kill a cancer without affecting tissue. And actually, what I'm specializing in these days is the concept of targeting multiple synthetic lethal nodes simultaneously as a way of taking down the cancer network. In any case, I want to show you this specific case that came up my way at CellWorks, where we have a patient who had 6 mutations and quite a disorderly copy number aberrations. There were 1,092 genes which were either deleted or amplified, and low terminal mutation burden, low indel, so not an immunogenic disease. If you want, I can show you what that looks like. Let me just put up a different document here, if I can. So I can show you what the CellWorks report looks like. Of course, now this is the challenge of navigating in my, am I going to be able to navigate my desktop? Here we are. Okay, so quite a long report because when you have 1,092 pathways. This is what happens. Okay, so we have a little summary here of the mutations. They were all loss of function mutations. None of them would be considered particularly actionable. And some of these things I have to confess that I would have to look them up, even though I specialize in this. There's a lot of obscurity, and which is why we need a tool like this, because there's no oncologist alive who could sit down and tell you what all these things do. I would challenge anybody to do that. Now, here's what the chromosomal aberrations look like. And by the way, this is what I call the elephant in the room, because even though the reports that we get from our companies, I'm happy to see that we have somebody from Boston Gene here today. Our reports they focus on mutations, right? And the reasons we do that because of the paradigm of treating oncogenes with tyrosine kinase inhibitors has been practice changing and really transformed the entire field of oncology in the last 20 years. The truth is, it's just the low-hanging fruit, because what's going on in cancer is this. Look, we got all of these pieces of chromosomes that are missing and extra copies. And this is really what happened. This is really where the disease is coming from. So what we're doing at CellWorks is trying to incorporate all of this information in the model to come up with predicting how this makes the cancer be the unique thing it is, and what impact this has on the treatments that are available to us. So let me go back to my other presentation now. So when we look at this patient's response to docetaxel, you're seeing the gene aberration on the left here, the effect that aberration has on the tumor, and then the signaling pathway that explains what's going on. And I'm just going to kind of run through this a little bit quickly. You can see that most of these things are resistant. There's a couple of sensitives in here. And it's interesting because we always see in these complex cancers combinations of things that are both sensitive and resistant. When we get to mitoxantrone, you start to notice that all the things that were resistant are now sensitive. In fact, a lot of the things that were causing resistance to docetaxel are causing sensitivity to mitoxantrone. And there's a BRCA2 deletion, which is not germline. It's a somatic change. There's also a ATM deletion, so DNA checkpoints are out, and we have the Fanconi pathway down here. It's also known to impact the response to DNA damaging agents. There's TOP2A amplification. Well, everybody knows this is a mitoxantrone and also happens to be a toposide and anthracycline sensitizing pathway. And so here is the therapy prediction index that is the main result of CellWorks. And it shows about a 4-fold difference between mitoxantrone and prednisone and docetaxel. And so if you hadn't done this biosimulation, you would be giving your patient docetaxel, and when that didn't work, you would give them cabazitaxel, which is also, by the way, not going to work in this patient for similar reasons. They're both tubulin targeting agents. And the drugs that really do work, which are the ones on top here, are not going to be assessed. And by the way, we find out that even though this patient doesn't have germline BRCA1 or 2, they have this really nice responsiveness to PARP inhibitors. Got a great response here to platinum drugs. And VP16 also hits topoisomerase 2A, and so you can get an additive effect. And radiation is sort of middling. Just to show you the signaling pathway impact of various pathways on olaparib, you can see that those are mostly all sensitive. And as far as radiation goes, I put this in here because people who are facing prostate cancer as initial diagnosis are essentially confronting the surgery versus radiation issue. And, at ESMO, there was actually a very interesting concept of using this new radio antibody, Pluvicto, in the upfront setting. Should we do that or not? I think that this kind of analysis can help us understand the relative utility of these various agents for individual patients. And so here, the same thing that creates sensitivity to the DNA damaging agents also creates sensitivity to radiation. On the other hand, the efficacy of radiation requires the intactness of the non-homologous end joining pathway, which has some deficits, and that's why radiation came out as somewhat inferior, let's say not as active as the DNA damaging agents. And so basically, mitoxantrone looks like it's about 4-fold superior to docetaxel. And what I've seen at CellWorks, by the way, is this happens to be true in patients with homologous recombination deficiency in general. We are giving the wrong treatment to these patients. This patient did not have a germline therapy and would have been missed. And so I think that's what makes it such a great example. He had some HRD, some Fanconi pathway, some compromised DNA checkpoints, and together all of those things, makes the DNA damaging agents and the PARP inhibition viable therapies, whereas the standard of care, docetaxel and cabazitaxel, really don't have much. And so what we do in evidence-based medicine is we do these huge randomized trials. They have to be huge in order to find tiny differences. And in fact, during the conduct of the trial, the investigators cannot tell who's winning. That's the problem of marginal therapy. You need a statistician and 1,000 patients to tell you that something's better. But here's the problem, in that even though it's true that, as I've illustrated here, B is better than A, right? Because it's got a bigger circle. It's only true for 30% of patients. A is equal to B. I mean, either drug could have been effective 10% of the time. A was in fact better than B, the opposite, 25% of the time. And neither therapy was effective for 35% of the patients. And so when you add up all of those numbers, what's generally true, that docetaxel is better than mitoxantrone, happens to not be true for 70% of the patients. So I think this is kind of an interesting, thing about the way we work in cancer, where many of our therapies are marginal, and the fallacy of marginal benefit is that we over-celebrate these trials. In the era of biosimulation, I think we can do a lot better than that by individualizing the treatment for specific patients. And companies like Boston Gene that are doing whole exome, and who have been extremely gracious in terms of sharing the raw data, the copy number, and also we're in this era of transitioning to transcriptome. I think they're really going to be leading the way here. And I at least am optimistic that I can do a better job for my personal patients doing this. But I also felt some obligation to get this news out to this group because I think you guys can appreciate this and are in a position to raise the consciousness about it. We're going to need to do a prospective randomized trial with CellWorks biosimulation in order to change medicine. And I don't know where the money is for that is coming from. And that's why, one of the things I see myself today as being involved with CellWorks is to try and raise consciousness about the fact that we're mistreating patients, where we are coming up with the exact wrong therapy. And since I'm a member of this profession, I have to admit, guilty as charged, because we've been stuck in the evidence-based medicine, fallacy of marginal therapies. So that's really what I wanted to say, and I'm happy to hear what you guys think about all this.
Okay. As usual, thank you, Michael. That was a good setup. Perfect brief timing. We already have hands raised. We'll use the hands raised feature to prioritize different questions and start the conversation here. Bob Germankin, you're up first.
Okay, thanks. So this is really very, very interesting, especially since I'm kind of actually there are people on this call who I've been reaching out to or have been reaching out. So a few questions. So when you have heavily pretreated patients, you have even more heterogeneity of the cancer. And when you're taking biopsy samples and running your biosimulations, you may do 2 different samples and get 2 totally different,
Right.
simulations. So how do you work with that?
That's a great point. It's one of the great challenges. The tumor heterogeneity increases over time because of genomic instability. And actually, I think that there's a relationship between the degree of genomic instability and our ability to run after this disease. And but I think from a practical point of view, allele frequency is something I use to try and sort that out. I had a patient recently, my latest patient is a fellow with liver cancer, and he has in his circulating tumor DNA 2 genes with mutation rates in the 30% range, and everything else is below 1%. So there's got 4 mutations below 1%. They're all like really bad oncogenic drivers. And it's got these other 2 in the 30% range. Well, I think I can neglect those 4 for the time being. And as I remeasure, as I follow this fellow and going forward, we may be selecting out those resistant clones. And so I think you treat the thing that is got the most, life-threatening impact in terms of disease burden, knowing that there are other clones you may not be treating and you're going to have to come and deal with them. It's sort of like a chess game, and you're not going to get this in one shot, and you just have to be ready for what's your mid-game strategy, what's your end-game strategy going to be. And, to adopt an attitude where you look at it as, in this dynamic way, as opposed to having some sort of algorithm fixed by the NCCN. Is that helpful?
Yes. So I guess my question is, how can patients, I'll be specific to myself, put that into practice? I mean, I'm scheduled for a bone biopsy next week. Michael Hensley at Boston Gene has been very kind, and they are willing to do sequencing. Also been trying to reach out to Dr. Apfel, who I got a message from his office about calling about functional testing, but the more data, the better.
Yeah. I'm working with Chris Affle myself, and I think he's a good guy, and he's also got a great team. Well, to really answer, I think depends on knowing, the individual patient's situation, how much bone metastasis, how many visceral metastases, what is the burden of disease in each site. What's the liquid biopsy show us in terms of heterogeneity? And the other thing about liquid biopsy is it gives you the sense of diversity of metastatic behavior in the body. So you might not get a mutation that's in the lung, for example, if you're biopsying the bone. And so, but you could see that with a liquid. So I like to do them both because I think they're complementary.
Okay. I guess in order to possibly have you work on I would do that through Boston Gene.
Yeah, I think Boston Gene is doing a great job, and I'm really happy to work with them.
Okay. All right, thank you.
Thank you, Bob. Next up is Alan Morris, also known as Shasta Pathology.
Yeah. Can you hear me?
We can hear you.
Okay, great. I heard you say the phrase synthetic lethal nodes, and I have to admit, I've never heard that before. Did your group invent that concept, or is that well known in molecular biology?
No, actually, I've invented that concept. Synthetic lethality is very well defined. It's been in the literature for probably at least a dozen years.
No, I'm not talking about that. I'm talking about
but the
synthetic
nodes.
nodes. Yes.
Yeah, exactly. So I've had generally what I am doing is when I get the results of a genomic analysis, I'm looking for how many things can I treat, and then I try and target as many as possible. And I've had some remarkable successes, including a case that's published in the Frontiers of Oncology last fall about a patient with relapsed glioblastoma, leptomeningeal spread with a terrible prognosis, who not only got a complete remission, but is alive 27 months later with no evidence of disease. And so why is it that she is doing so well? And I think that essentially her situation was unique in that she had about a dozen synthetic lethal nodes that were all successfully targeted. And so this idea of network targeting combination therapy came to mind. I thought I invented that. Turns out that was invented by an Australian mathematician, Robin. Her last name will come to me in a second. In any case, it's the idea that the network is kind of like the Paris Metro or the London Underground. If you've been to those places, perhaps, they have such amazing redundant systems so that if one station goes down, you take a different line and can go around. And, normally what we do in oncology when we have a network like that in cancer, we drop a bomb in the middle and hope that that's going to do it. And that's not really the way it works, because people will just say, okay, well, you have to take a different train and go around. That's what cancer is doing. On the other hand, if you create a small explosion in multiple stations, key stations along the way, whether those are synthetic lethal nodes or oncogenic nodes or master regulator nodes, I think the network collapses. And if you can hit enough of those things in the network, it doesn't recover. So if you hit Achilles in the heel, he can still sit up and fire his arrow at you. But if you hit him in both heels and both elbows, he's not going to get up. That's the idea. And so the idea that the synthetic lethal node is an important node, I think, is the key for how we're going to go forward in combination therapy for cancer, because all of these toxic treatments that we have, you get to the problem of polypharmacy. Eventually, the human body can only take so much. But if you're doing synthetic lethality and normal tissue can handle things, then you can stack therapies one on top of each other without really ever getting anywhere near the MTD. That patient I just mentioned, who had the great remission, she had no toxicity. It's even more remarkable. So I've written a couple of publications about this last year. If you're interested, you can email me and I'll send them over to you.
Please do. Yeah, Roger Royce is requesting that in the chat. We'll do that.
I have a guess about your synthetic lethal nodes, and I'd like your comment on it. I'm guessing that your synthetic lethal nodes are largely the DNA repair pathway ones. Are there other ones, or is it mainly DNA repair pathways that are the synthetic lethal combination nodes that if you throw them into the Paris metro, such as like if you got an ATM and a BRAC and a check or something, you hit all those and then you get the synthetic kill. Is there other pathways?
Well, you're absolutely right. I mean, we already know about the HR pathway, right?
Yeah.
And the HR pathway kind of includes the Fanconi repair and also the DNA damage checkpoints. But also nucleotide excision repair is extremely important for certain types of drugs. For example, the nitrosoureas in neuro-oncology. And it's actually turns out that normally a p53 mutation, for example, is a bad thing because we rely on p53 to trigger apoptosis under DNA damage. And so if your p53 is knocked out or functionally disabled, then you're not going to trigger apoptosis. But on the other hand, you need p53 to express XPC for nucleotide excision repair. And if you don't have that, it turns out certain drugs, including platinum agents, are much more effective. So it becomes another one of those things where the mutation for one drug could be bad, but for another drug could be good. And so I try and be extra aware of those. There are other things too besides the DNA damage repair, and one of them has to do with energy. For example, if you have STK11, you're going to have difficulty with triggering autophagy for energy production. And one of the things I did in this GBM case was to target ATP synthesis at the level of both the ATP pump on the cell membrane using digitalis and metformin targeting of complex 1 in the mitochondria. By the way, this is the ultimate in translational medicine because, all my colleagues say, oh, this is speculative, there's no evidence for this. They're absolutely right. No one has done clinical trials of this stuff. But if you read enough molecular biology, these are the things which PhDs in the labs, they've been spending their whole careers on these things, and they're sort of like taken as fact. So it's not speculative in the laboratory, it's speculative in the clinic. But I think that it may be speculative, but it's a light. It illuminates an otherwise dark room. Another area I think is interesting for lethality, synthetic lethality, is in the area of oxidative stress, because oxidative stress has to be extremely tightly managed, otherwise it kills the cell. And it seems that some oxidative stress is actually oncogenic because it activates oncogenes and can disable tumor suppressor genes. But if it gets out of hand, then it's going to lead to cell death. So the cell's ability to defend against oxidative stress is usually upregulated, but it can be targeted. And so that's another area that I'm always paying attention to these days.
Thank you. Frank Notehaft, you're next.
Hi, thanks for the presentation today. Very, super interesting approach. I'm a, kind of molecular biologist, bioinformatician, depending on how people look at me. And was kind of curious, definitely relate to your point here about a lot of this being kind of known in the molecular biology literature, but not in the clinical space. I'm kind of curious, have you tried to do any sort of like synthetic trial replication studies to see if you can like explain some of the trials where you see, just tremendously variable, strong response, non-response? For instance, like one that kind of comes to mind is Pembro, especially as Pembro has moved into like TMBH and MSIH, where you see some patients who have tremendous response and other patients who don't. Something like the model that you're looking at feels like it could be very insightful in trying to explain what are the drivers of response, non-response in a case like that. So I'm just kind of curious if you've looked at doing any sort of synthetic validation that way. Obviously, it wouldn't replace like, full clinical validation, but it would be an interesting study in the middle.
What we've been doing, and you're going to see some publications in the near future in non-small cell lung cancer, is to do prospective, retrospective studies where the outcomes are known.
Yeah.
And then we get the data in a blinded fashion, the genomic data, we put it through the model, and then we predict outcome. And we're actually working with the statistician Drew Watson. Drew is the person who invented Oncotype DX for breast cancer. And he's been a delight to work with, where he can take a given parameter and come up with a way of weighting that with exponents according to its relationship with survival. And then in the end, create an equation which will tell you, whether or not immunotherapy is going to work and what the patient's survival will be given a certain set of inputs. And we are addressing the clinical conundrum in lung cancer about whether or not you should give chemotherapy simultaneously with. And actually, it seems that if you have, if CellWorks gives you a high score, then you don't need chemo. And if you have this low score, then you should get chemo. And that's independent of TMB. And, in the statistical analysis has a huge chi-square compared to TMB and PDL1. So absolutely. And, a lot of the intracellular pathways that we come across, when I was glancing through that list of things I showed you in that last case, the patient had PTPRD deletion. Mm-hmm. And PTPRD is a phosphatase that reverses STAT3 phosphorylation. And STAT3 basically poisons the tumor microenvironment and reprograms macrophage to cause, M2 polarization, which quashes the immune response. And so you've got to deal with STAT3 in that patient if you're going to give or consider giving pembrolizumab. So I think that not everything is actionable, A lot of the, for example, beta-catenin is a problem in that it's changing around the cytokine milieu of the tumor microenvironment. And, how are we going to overcome that, So we don't have drugs for everything, but we have one of the things that I've started doing almost out of the sense of therapeutic necessity, or what I call the therapeutic imperative, is one of the things that gets upregulated is the AHR, this aryl hydrocarbon receptor, and that activates TDO2 and IDO1, which are the things that make for quashing the immune response. And it turns out that you can block this with a substance used in traditional Chinese medicine called tangenone. That's pretty wild. And so I've become, knowledgeable about how to do that. I'm giving tangenone to my patients who have that biology, because otherwise I have no hope that pembro is going to do anything.
Mhm.
It usually doesn't for most patients, right? Unless they have very high immunogenic cancers or something else. The MSI high you mentioned is a little bit easier question, because I think in that situation, the MHC1 apparatus gets knocked out, or it's the choreography that gets it inserted into the membrane is knocked out. When we look we get this fancy copy number analysis from our NGS associates, we find out that, HLA genes are being deleted. And so I guess those are not too useful for antigen presentation. So I think there's lots of different things to learn in each patient's case, and we try and make the best of it once we get that knowledge.
Sounds good. Thanks for the answer.
Ian Lewington.
Thanks, Michael. Good morning from New Zealand.
New Zealand, oh my.
Yes, yeah, international audience. I'm a bit like Rob, I guess a patient perspective question. So when I was diagnosed de novo stage 4 advanced, I ended up with docetaxel and ADT and had a favourable response. Obviously didn't eliminate, and have a relatively low tumour burden at the moment, with the light looking like I'm starting to get resistance sort of 2.5, 3 years later. I've had liquid biopsy, which shows very low mutation levels now, even though I had TP53, P10, and tested, and oleopara wouldn't work. Is the testing you do useful for someone like myself, where you've been fairly heavily pre-treated? You're looking at next lines of treatment, what next sort of ADT you should use? Is there a different chemo combination? And at what point should I be letting the tumor burden get higher before you tested, or when would you test?
Yeah, I think it's a practical issue, and it's going to depend on reviewing the radiology and seeing, is there anything we can see to put a needle into and get a biopsy from. The CellWorks model is not informative for what kind of ADT you should be on, and so I would simply explore those options in the way we usually do. We have a bunch of very effective drugs that seem to have slight differences in their mechanisms of action and can be used in sequence, and I'm not sure that any one sequence is better than another. As far as chemotherapy, I would definitely be interested in that, but the answer is, for what CellWorks is doing, we really need a high-quality NGS. And to get a high-quality NGS, you need enough DNA in order to get a copy number. And so, I would do a liquid biopsy now, and I would definitely be interested in scanning your body thoroughly, and then trying to say, is there anything that's useful to biopsy? Of course, prostate cancer is unusually challenging because it has its predilection for bone. And, you know, it's I think, much less certain what the outcome will be when we put a needle into bone compared to putting it into a liver metastasis or a lung metastasis. So, you know, some of it depends on the skill of the person who's doing the biopsy. My radiologist spent 2 hours with a patient the other day because she was sure she didn't get it, even though she could see radiographically the needle was in the right place. She knew that there wasn't enough tumor in that specimen, so she kept at it. 2 hours, imagine. So I think that, you know, that's a great doctor. And not everybody has those kind of, you know, that bandwidth to be able to do that kind of thing for a patient. So, you know, in the end, I would talk with your doctors about this and ask if you could see the scans yourself.
Yeah, look, I've seen the scans, and yeah, I do only have bone mets, no organ involvement, and largest met has only got SUVs of about 2 at the moment. So it would all have to be off the bone.
Can you see them with an MRI scan?
I'm not sure. I've only had PSMA scans.
Yeah, I would do an MRI next and see if any of them are targetable with a needle. Especially sometimes they can be sticking out of the bone a little bit or more towards the surface. And those are the ones that I think are more interesting to go after.
Thank you very much.
Sure.
Vanessa.
Hi, I am a GBM caregiver, and I know we have a couple GBM patients and caregivers on the call today. So I wanted to ask, your approach is pretty novel. So where do you see it fitting within the continuum of care, where, you know, in GBM we're giving TMZ to MGMT unmethylated patients at the newly diagnosed stage, and then we have no standard of care at the recurrent stage. So where do you see your approach fitting into kind of our care process?
Yeah, GBM is a special subject. I became a neuro-oncologist after, well, around age 60 or so, because of certain very special patients that I was involved with, actually, who changed the trajectory of my career by getting me involved in what I'm now doing. And so I'm especially interested in trying to do something with this group of patients. I think the more you know, the sooner you know it, the better. And I would encourage everybody to get the deepest possible molecular diagnosis as soon as possible with GBM, and then to use whatever tools you have. CellWorks, I think, is a pretty extraordinary tool compared to, because, you know, if you're going to have hundreds of chromosomal abnormalities, and you give this to a practicing oncologist, and they just kind of, you know, throw their hands up or shrug their shoulders and say, "What do you expect me to do with that?" And they don't know the biology. Even if they know a pathway or maybe a few pathways or maybe a couple of, you know, for example, DNA repair has about 497 genes in it. That's an awful lot of genes, right? There are more than 2,500 transcription factors that work in groups. They compete against each other to upregulate and downregulate gene expression. Wow, it's an overwhelming complexity in our cells. And so I think that in order to take the next step in human history, as far as understanding these complex diseases go, we're going to need an AI level kind of oracular tool that can show us the complex proteogenomic networks that we're dealing with. And then the question becomes, how are you going to take out that network? And another analogy that I have is thinking about the way the military is conducting things. You know, we hear these sort of things now that they're going to hit the communications, they target the radar, they get the fuel depot, they, you know, then they hit the airstrip, and then they, you know, they come and they attack, you know, from 5 directions at once. So it's an overwhelming, they're attacking all the network of the enemy. And if you do that effectively, they're not going to recover. There's nothing to recover from, right? That's the concept in cancer. And then it becomes an issue of how can you do that without killing the patient? And that's why I think the concept of synthetic lethal targeting, where you can stack multiple therapies simultaneously without impacting normal tissues much, is the key forward. Now, you mentioned glioblastoma. I have several case reports I'd like to send you about.
I've read the one you mentioned, the
You
LM.
did? Okay. There's another one in the relapse setting. And I have some other ideas, by the way, how to keep going with this disease. You know, the standard of care is a very weighty thing. If you don't do it, people think you're a bad doctor. There are 10% or 13% of patients with NGMT unmethylated disease who do seem to have a benefit from that treatment. If you can understand who's those people are and not deny them a drug that works, that's a virtue. CellWorks can do that. And in fact, one of my projects is for this year is to try and get that data out. So on the other hand, we have to develop a will as a profession to stop doing things that don't work. We're wasting time. We're causing toxicity. We're harming patients. And we have to take a strong look in the mirror and develop the will to stop giving treatments that don't work. You know, I established this rule in my practice a number of years ago, and I can tell you it makes me a radical. The simplest thing about not doing things that don't work. So, but I also believe in a lot of the papers I read. And if you don't read those papers, then you never think about these things. So most of my colleagues don't read. So.
Thank you. And I really look forward to seeing that research on which unmethylated patients respond to TMC.
Great. Yeah.
Brian's up next.
Hi, Dr. Castro. Great to see you. So my question is really around multi-omics. So right now you have a model that's incorporating genomics and transcriptomics, and it has a certain amount of, you know, hopeful predictability, right, in terms of predicting drug sensitivity. But that's only part of the equation. You know, as we've kind of talked about on this call, prostate cancer is a cold cancer, and the immune system changes over time when advanced prostate cancer patients are exposed to multiple different treatments. So I've worked very closely with Boston Gene to look at how my tumor has evolved over time. I don't know if you're familiar with their tumor evolution report, but it's pretty awesome.
Yeah.
And part of that is looking at the immune profile, right? So I've gone from an immune desert when I was first, when I had my first surgery, prostatectomy in 2016, to a fibrotic immune profile. So how do you kind of like factor in these additional data sets into your existing model in terms of understanding the likelihood that a patient is actually going to benefit from any one of these combinations of drugs? And where does proteomics fit into that? Maybe I'll start with, we can start with maybe immune profiling and then maybe get into like proteomics or other analyses that can add a higher degree of predictability to your model.
Yeah, well, these questions you guys are giving me are really terrific questions. You know, I think the evolution of the tumor microenvironment is a really fascinating thing because it's reflecting the evolution of the cancer under some therapeutic stress. Fibrosis normally, to a large degree, is mediated by TGF-beta. There may be other things also, like CXCR4. But, those are potentially targetable. And TGF-beta can be targeted with losartan, which is an ordinary antihypertensive that's actually been studied in pancreatic cancer. And in the lowest dose, 25 milligrams, it doesn't cause hypotension. In stage 3 pancreatic cancer, for patients who were unresectable, they had a higher down staging and higher R0 resection rate by adding losartan to FOLFIRINOX. And unfortunately, in stage 4, whatever is going on in pancreas is already beyond just giving losartan, okay? But I do think it has a role for blocking TGF-beta. And TGF-beta is practically ubiquitous in human cancer. It's almost always involved, and it is a real challenge. So I use losartan sort of very liberally and without really too much problem. As far as this transition to proteomics, everything we're doing with genomics is really trying to figure out what's going on with the proteins. And the idea of DNA goes to RNA goes to protein is actually, it's a lot more complicated than that, right? A lot of things that they have no problem in the world of DNA, like MYC, oncogene, for example, is a dominant player because its ubiquitin turnover is downregulated, and it gets sustained in various ways. So that even if it's not mutated and if it's not amplified, it still takes over the cell. And I think that the more we know from the world of proteomics, the more humble we get about genomics only. This company, they were just acquired. It's funny, I use them, but I can't think of them right now because we're in prostate land. But their profile is really useful in breast cancer. And 10% of those patients who are HER2 negative are HER2 activated. And so we have false negative tests, because when you treat those patients with anti-HER2 agents, even though they don't have HER2 amplification, they respond. That's a major discovery. So I think there will be, more and more. Now, the transcriptome is much closer. And what I noticed from looking at the transcriptome is there are a lot of things that you see in the genome that you don't have to pay attention to. I had a case recently, it was a GBM case, and there were like 15 tumor suppressor genes that were missing. They had mono deletions. And how am I going to deal with that? I mean, it's too many things to deal with. So in the transcriptome, 14 out of 15 had normal expression levels. So the normal allele was compensating. And so that's an extremely powerful tool for narrowing the mission or focusing in the right place. We'll have more abilities.
So I guess, is that to say that proteomics would trump anything that you're doing with DNA and RNA, or do you need the full model? Because I am one of those patients who actually has the whole central dogma, DNA, RNA, and proteomics.
Yeah. The problem is,
so
I don't think
as
we have.
as I'm trying to make a decision, right? Where do I place my weight? Do I look at all the data in its entirety to piece together something that is more than just proteomics? Or do I just say,
I
hey,
think
what, proteomics are, that's the gold standard.
I think proteomics is super important when we're thinking about developing CAR T cells or CAR NK cells, because you want to be sure that antigen is sitting on the surface. Similarly, for the ADC, the antibody drug conjugates, which are using the antibody as a smart bomb for delivering the chemotherapy to the right place. It's nice to know that that antigen is really sitting there on the surface. But I'm not aware that there are any proteomics that are in the 5,000 protein range. I mean, I understand it's about 5,000 proteins that runs a cell. And so, but we can get the entire transcriptome. I wish we get the microRNA as well. And we can get the entire genome. And so I just don't think the proteome is there yet to replace those other things. And I don't think we have enough experience with the transcriptome yet as inputs. But that's a kind of project that we have this year at CellWorks. And I'm sure other people are working on that too.
I'll just add a point of validation that nowhere in my DNA or RNA did HER2 show up as a gene that was reflective of my as a cancer driver. But in my proteomics, I had a moderate expression of HER2. So there doesn't necessarily seem to be a direct correlation based upon the data that we have with DNA and RNA to proteomics. So I think just a point of validation, right, that understanding that relationship maybe is still very.
It's a great point. And for those of those people on the line who don't think about these things every day, if you do something that affects the turnover of a protein, you can prolong its half-life and stability in the cell, and it can take over in an oncogenic manner. And you can have something that's, the turnover of HER2 from the surface of the cell, where it goes into the lysosome for destruction, is a heavily choreographed process. And if you either accelerate that or block it, then HER2 accumulates on the surface of the cell. And by the way, immunohistochemistry is proteomics. It's 1930s proteomics, but it's still, is that Shasta pathology up there?
Yeah. So yeah, exactly. Yeah.
I mean, it's old, but it's good. Still, it's still true. And that's why, you can, if the pathologist can see it, wow, that's a target.
Yeah, I'll turn it over to Alan. I can see he's chomping at the bit here, and he would be the next person to follow on this trajectory.
Let me just do a quick time check. We were at the top of the hour. Dr. Castro, how's your time look? Are you able to go a few more minutes?
Yeah, I can go a few more minutes.
Okay. And I wanted to interject a question because, but I was waiting because it's going to be Alan's second, so I get to throw one in. I'd like you to, for a layperson who doesn't understand microbiology, just really helicopter up to the big picture and say, where does, at various times you've called this complex, speculative, just where does this sit in its maturity? Is this in the hype cycle? You mentioned AI and immediately, there's a question about hype cycle. We've had people on this forum before who've said that the modeling of biology is incredibly complex and quite difficult with a fraction of the numbers of variables which you've cited. And so, who is this for? Really, it's really a question for what patient should be availing themselves of your services, given the, I guess, early stage nature, the research lab nature of what you're doing.
Thank you for the complex question. I mean, I think it does come down to a matter of philosophy. And what I do is not very comfortable for many people because it is outside the box. It is vanguard medicine. It is taking off the training wheels and trying to figure out a solution to the problem, since I'm in the twilight of my career, I've decided I don't want to just continue doing things that don't work, right? Because I made this pact with myself to stop doing that and to find solutions. The more I do that, the more I seem to be able to be effective. But it does mean that you make yourself a guinea pig. And you can either walk the plank as a patient, forgive the crudity of this concept, but I do see it that way as an oncologist, that, getting metastatic cancer is, it's your ticket to the next world. And you can either go with a standard of care and the NCCN and consensus medicine, where you can ask, is there anything else that's been learned about cancer that could help me? Is there anything that anyone could learn about my tumor that I can leverage as a therapy? And the answer to those two questions is a resounding yes. The forefront of cancer research is decades ahead of the clinic. And unfortunately, most of what is being learned by the great public investment in cancer will never be translated into therapy, because that translation is driven by one thing, and that's profit. We live in a society where the first words are, show me the money. And if you can't, and then it's not investable. You need intellectual property, you need development plan, you need the right team, you need clear steps to getting something that can generate a $20,000 to $30,000 a month per patient, charge. And then you're going to develop that therapy in isolation, as because that's how things get on the market. But that is not the way to treat cancer. Because once you get the view that I've gotten from working at CellWorks about the complexity of the disease, you can see how meaningless it is, most of what we do. I mean, the current drug development efforts are so naive as to be laughable. And, there's an old saying that people who live in glass houses shouldn't throw stones. And so I have to be careful how I put these ideas out in the world, because it's going to offend a lot of people. But the truth is, if you're the patient, you don't have time for all of these shenanigans, right? And so I think the person who should work with me is somebody who is, number one, believes in science. Number two, is willing to go outside the box and even be in an uncomfortable relationship with other doctors who say, there's no evidence for what Michael Castro is doing, and so therefore I wouldn't do it. But one thing that's happened to me as I've gotten older is I know I'm close to a number of people who have passed away of this disease, including friends. And, it cuts pretty close to home, even though I haven't yet gotten a diagnosis. I realize it could happen. And it's 50% chance, in fact, that the average male is going to get a malignant disease. So I always put myself in the patient's shoes and say, knowing what I know, what would I do? And I've been extremely fortunate to be, practicing oncology at a time in history when I've seen people who are, 9/10 into the grave have complete remissions and go back to meaningful lives. I've seen it multiple times in my career. And so I have, and with the CellWorks biosimulation tool, I feel, even though, as the old, there's an old thing, all models are wrong, but some of them are useful. I think that this is true of CellWorks is true as well, that this is an incomplete model. But it's a lot more than anybody else is working with, and it's so much further ahead than any clinician can do on their own, that, I think looking for its utility is interesting. And what got me interested in this whole thing to begin with was when I would get a report from CellWorks, I would see things that I simply didn't know before. And it was like, oh, that's interesting. What do I do with that one? And but I do think it comes down to philosophy about how you want to be treated, and where your comfort level is. Remember in that movie, The Matrix, that scene where he comes up with you can swallow the red pill or the blue pill. If you swallow the blue pill, you wind up in your bed knowing you're receiving the very best care possible from the very best doctors. And if you swallow the red pill, then we're going to show you the way it really works. I think, if you're at Stanford or UCSF or, City of Hope and, all these great institutions, you're getting that blue pill treatment. You're getting consensus medicine, NCCN guidelines, or somebody's idea of, clinical trial. Clinical trial may or may not be useful for you. I'm not against clinical trials at all. I'm for those, but not all of them are good for all patients. And so one wants to be shrewd about it. That was a long answer. I'm sorry if it was. I hope I answered some of your thoughts.
There are a lot of nuggets in there, Michael. Thank you. Alan and Frank, and then we'll hopefully wrap it up here. Alan, are you?
Yeah, I'm ready. I'm ready. Okay, Dr. Castro, I'm blown away. One question I'll ask you, which you'll not know the answer to, is how many clinicians are also molecular biologists? How many people actually wear those two hats in a real way? And I suspect that you are one of the few, but maybe I'm wrong.
You're right. I work with a guy. Actually, pathology now has molecular pathology.
Exactly.
They're the first ones to do it. And I've been arguing to people at ASCO, you've got to create molecular oncology as a certification.
Exactly. And there is molecular pathology as an actual certification, but I bet you, and I've done the math on it, there's only about 300 board certified molecular pathologists in this country. And one of my bugbears in my brain is that there has always been a chasm between the closest thing to science that medicine is, which is molecular biology, and the closest thing to clinical medicine that molecular biology is, which is molecular pathology. That's an absolute chasm. And you need a hybrid person like yourself to see it.
Yeah.
And not only that, you need a hybrid person like yourself who then has AI to try to get at the stochastic system that the complex molecular biologic system that is us as humans to try to even get at. I applaud you for being the inventor of the word synthetic lethal node. I want to give you some crazy thought. There's a primordial molecular node, and it's NFKB beta, and I'll tell you why. They have done knockout. This is one of the original molecular biology experiments in vis-a-vis David Baltimore back in 1977, discovering NFKB. It's knockout mice. For those people that don't know basic molecular biology, how do you figure out what a gene does? They did knockout mice on NFKB, and I'm sorry, I have to give people background about molecular biology. NFK beta was one of the first tumor necrosis factors. So it was discovered in the context of immunology. But when they did knockout mice experiments and they knocked out this gene, unbelievably, you didn't have lymph nodes. No lymph nodes. I mean, that's profound. So this one molecule, you knock it out and the person doesn't have lymph nodes. The lymph nodes are the organ, one of the, aside from the thymus, the lymph node is the major organ of our adaptive immune system. And the other unbelievable thing is that denosumab, which inhibits rankle, actually, oh, and then the knockout mouse thing also is they don't have bones. So you have this you are the inventor of the synthetic lethal node. Well, there's a primordial molecular node in the adaptive immune system, which is the holy grail for the immune cancer theory.
Fascinating.
Have you heard this before, or am I inventing this?
I think you're inventing it. I mean, I haven't heard. I mean, I'm obviously NF-kappa B is a big issue. You know, you need NF-kappa B in your functioning immune system in order to mount an immune response.
It's primordial.
On the other hand, NF-kappa B is a major driver of cancer, particularly the lymphomas, myelomas, but also solid tumors, because it happens to be downstream of AKT. And the PI3 kinase AKT pathway is dysregulated in 90% of every disease I treat. So at least maybe 100%.
Yeah. So there, because of this nexus, there's actually a term called osteoimmunology, and it is still in the dustpan of history. It's there. I don't think people are driving on that. I'm not a molecular biologist. You're this rare bird that is bridging molecular biology and molecular pathology. Do you know if osteoimmunology is taking off or not? Which is interesting because, crazy enough, prostate cancer of all the cancers is so bone-centric. I mean, that's telling that it's so bone-centric.
Yeah. I agree. I think there must be something in that.
There's something in there, and it's the curiosities. Like, for people like you, if you drive down on the curiosities, you're going to make a breakthrough. And I'll tell you, I pose these curiosities to expert prostate cancer experts, and they don't even attempt to answer it. They don't even attempt to answer it because it's not the eye sees. One of the first things I learned in pathology from one of the original Virchow school pathologists was the eye sees what the mind knows, and their mind doesn't have it in there for their eye to see, you know? Never mind. I'm sorry. I'm going. You know what? I want to bring this down to earth because there's a patient here, Robert Dermankin, who asked you about bone biopsies and whether or not you could extract your magic out of it. And I bring him up in particular because he'd be a fascinating case for you. And the reason he'd be a fascinating case is he's one of the rare prostate cancer advanced patients who is mismatch repair positive. And also what's fascinating about it is he did try Keytruda, which I thought would absolutely work. And he thinks it's failed. And the reason he thinks it's failed is because his clinicians think he had pseudo progression. So there's this concept of pseudo. This is a radiology problem. So I'm bringing, now you have to put your clinical hat on. I'm sorry. I know this talk is about molecular biology, but now we're putting our clinical hat on. So he tries, he's an ideal candidate.
Alan, can you close it out? Because
okay.
we're out of time.
Okay, I guess I'll just ask a generic question. What do you know about pseudo-progression versus progression vis-a-vis radiology resist tests trying to get at whether or not you are accelerating by virtue of Keytruda? Especially vis-a-vis a mismatch repair patient.
Pseudoprogression's very common, and it's a conundrum in radiology, and it can't be solved in radiology except by doing serial scans over many months. We need the circulating tumor DNA assay, either an MRD test like Signatera or the 1,000,000-to-1 test called North Star Response, which is a fantastic technology. They can tell you the difference of 0.1% difference getting worse or better with their technology. So if you guys aren't familiar with Billion to One, I would turn you on to them.
Look into it. Frank, last question.
Sure. I'll keep mine just really short. And I realize this may be one that has to follow up offline. You know, as somebody with like a network biology background, especially like with curiosity working on transcription factors, you know, one of the methodological challenges that we faced with these very large, many nodal models is overfitting. You know, because fundamentally, you know, if you look whole genome plus whole transcriptome, you know, you're looking at 20,000 genes times 2. You know, I'm kind of curious, you know, how are you all approaching that problem? Because that'll, you know, that's kind of always been like a really confounding issue with these, you know, whole exome, whole transcriptome, cancer predictive models of progression.
Yeah, I don't know if I'll be able to answer the question.
Oh, no worries. It may be offline with
it's
the
better maybe to share the question, but you know, I think the ability to think about cancer from a hallmark behavior perspective, like things that are causing proliferation, things that are upregulating DNA repair, things that are changing the apoptosis pathways. You know, those that the histogram index that I showed you before, what does that really represent? That represents the net cell number. Net cell number is, you know, kind of the difference between things that are growing and still alive, you know, as after cell death, right? So you can have various ways of getting there. And, you know, what we're hope to do at CellWorks ultimately is to validate that. And, but the model itself, I'm not sure we can do it without somebody like Drew Watson, who's going to come in and then give us weightings for specific individual phenotypes. When I'm talking about phenotypes, I'm talking about 130 different pathways. And those pathways are themselves complex. And so what you're saying is absolutely true. It is a level of complexity that taking on is, I can tell you the company has been doing it for 15 years. It's been in incubator mode in Bangalore, India, where the cost of hiring a PhD is about 25% of what it is in Silicon Valley. Mm-hmm. So, you know, it probably wouldn't be possible to do this kind of development in the US because of that. So, but ultimately we'll need to have some validation. But I think if you can follow, you know, if you believe in, you know, if you can understand one pathway and you can understand another pathway and you start putting it all together, it's the integration and the predictive value that becomes tricky in the end.
Oh, sure. Yeah. But even, you know, even like if you look at, you know, let's just think about like transcription factor biology, like you're looking at like binding complexes of multiple transcription factors where like,
yeah,
you know, predicting
plus the promoters that affect individual transcription factors is a whole other level of complexity that we haven't even sorted through yet.
Right. Exactly. So it, you know, it becomes a very large
transcription factors are at the end of signaling pathways that are also complexly dysregulated in the tumor. So, I mean, it's mind-boggling.
Right. Yeah.
That's why, you know, finally, to have the network view of cancer, I think is a completely revolutionary and modern view of what the disease is. Because I can say cancer is a complex proteogenomic network. You say, people, yeah. Tell us something new, right? Well, but, you know, here's what you're saying is exactly what I'm talking about. It's really complicated. And until we embrace that, this idea of this individual thing that's feeding back on itself. In fact, I don't know if you guys, do you have 2 minutes to see something else I created? that kind of explains this concept. I would love to show it to you. It's an animation that I had created to explain this concept of what is cancer. And let me see here. And I would just share my screen again. So I have to come back to Zoom. Okay, so what I've done here is to make a little diagram of putting the transcription factors in the periphery and the signaling pathways on the inside. Here's NF-kappa B, by the way. And just to show you, this is what I began to understand from working at CellWorks. Now, what you're seeing here is I've listed on the right side something that has, redundancy, convergence, crossover, reciprocal stimulation, feed forward activation, and I'm sorry. And essentially, this makes the tumor network completely autonomous. And once it gets going, there's nothing that's going to stop it unless you have some particular therapy that can take down the key nodes in this network. And so, but I think that, from the point of view of thinking about how should we be treating cancer, we have to start with an understanding that looks more like this, and not simply take one single oncogene and say, okay, how are we going to block that? And what disease should we develop this drug in? And the way we're doing it now is so silly and naive that I think that, we have to get smarter than that.